Well, good afternoon, everyone. It gives me a great pleasure to introduce the distinguished lecturer speaker, Professor Mu-ming Poo. I think most of you know him, so I actually will give a very brief introduction. Professor Poo got his bachelor degree in physics from National Tsinghua University. And he got his PhD in biophysics from Johns Hopkins University. And after his postdoctoral training at Purdue University, he served as faculty in a number of universities in the US. First is UC Irvine. Then he moved to the East Coast and served as faculty at Yale, Columbia, and then moved back to the West Coast, served as faculty in UC San Diego, and settled down in UC Berkeley in year 2000. He is also the founding director of the Institute for Neuroscience in Shanghai under the Chinese Academy of Sciences. And what he has accomplished in mainland China, I think, is remarkable. In particular in the field of neuroscience, and he has built the Institute for Neuroscience to really a showcase laboratory in mainland China. Professor Poo is well known for his seminal contribution to our understanding of the signaling mechanisms that underlie nerve growth and guidance. Also for his contributions in our understanding of the basis for learning and memory, and also synaptic modulation by neurotrophins and electrical activity. Because of his significant contribution to science, Professor Poo is a member of many academies, including the US National Academy of Sciences, Academia Sinica. He's also a foreign member of the Chinese Academy of Sciences, and more recently a founding member of the Hong Kong Academy of Sciences. And he has also been extremely active in the establishment of HKUST since the founding stage. And I want to add that I'm here because of him, because he recruited me to HKUST without himself being here. So that's a remarkable achievement. And because of his scientific accomplishment and also his contribution to HKUST, he received the honorary doctorate degree in science from our university in 2014, less than 2 years ago. So he's visiting us this week, and he will give 2 talks. There will be a talk on Thursday as well. It will be more scientific. For today's talk, he would like to share with you his view on research innovation. And I'm sure many of you in the audience, faculty and also students, will be inspired by his talk. So please join me in welcoming Professor Poo.
Thank you, Nancy. My great pleasure to be back here to give a few lectures, and there will be more after this week, I hope, in the coming year. I'm as a visiting professor for the Institute of Advanced Studies. I want to have the opportunity to talk not only about science, which I love to talk about, but that in my own area of neuroscience, I would also like to discuss things in general with especially students, because my job as a scientist is also involved training students in doing science. I would like to tell you, summarize my impressions, my thoughts on many aspects of research today on the innovation aspect, because innovation seems to be the key words you hear everywhere. When you go into this campus, you have innovating today, imagining tomorrow. I saw that big if you look at newspaper, the worst innovation in any newspaper in China is the highest frequency. So now the question is, what is innovation? How do you become innovation? This is a big, interesting question, general question that we all want to ask. So let me today tell you what I think innovation is about. I would like to explore the nature of innovative research. What is the characterize innovative research? Then I hope to be able to talk about how to learn to be innovative. This is based on the idea that innovative ability can be learned. It's not intrinsic. You are not born with innovation, capability of innovation. It's an ability to learn. And in this, now you would touch upon the origin of innovation. Where does the innovative idea comes from, and where's the imagination comes from? Now this is very tough, very mysterious questions that I don't claim I have answers to it, but I think it's interesting to explore. Then I hope to discuss, if there's time, about the different type of innovative research, how to do those research, this including the exploratory research, right? Or hypothesis-driven research. So there are different types, and how innovation is involved in these different types of research. So let me start with the nature of innovation. So innovation means something new. The first characteristic is novelty. Right? There are many forms of novelty in science. You can find new phenomena, you discover new phenomena people have not seen before. That's innovation. You can propose new hypotheses, new theory, a new way of analyzing the natural phenomenon. That's innovation. You can invent new methodologies and technology. That's innovation as well. You can even just discover new evidence for an old hypothesis. That's also very important, right? Even though you didn't propose the hypothesis, but the evidence is new and important. And also, you can maybe even, you didn't discover phenomena, you didn't propose hypothesis, but you find a connection between this phenomena that is new, that nobody has seen before. That's a form of innovation. So all these are what we want to do as a research scientist. I say to the beginning graduate student, how do you do one of this? Now, certainly you are trying to discover new phenomena, but the easiest thing, the first step, I ask my graduate student to be innovative before they have a new discovery of phenomena, or before they propose new theory, which is very difficult for a beginning student, is to develop new way of analyzing data. It's the third form, I think, that one can start with. You collect data. So the natural reactions, you have a set of data. You go through the paper, what has been done in previous papers in your area, how they analyze the data. You follow the same way. That's all my graduate students do. And I come back, they would plot the same plot that's been published. So I say, well, why don't you invent a new way of plotting the data? Find a new way of analyzing your data. That's new. Other people have not done before, but with the same data. Right? And if you think about it, there are all the data can be analyzed in different ways. So you have to, there's a room for innovation, just for the analysis of data. Well, that's the first thing a graduate student can do when they have the data in their hands. Right? So that's a thought. Even now, as time goes by, you will be able to see new phenomena. Maybe you can make even more novel findings. So not everything new is important. We talk about innovative research. We don't want to invent something that nobody cares. Right? You don't want to invent the 11th way of killing a cat. No, nobody cares about that. Right? You want to be something significant. So what is significance? How do you decide the problem that you're working on is significant? This is related to the question, what do you think, what does your peers in your field think is important? Now, do you take the opinion of your colleagues and a peer? When everybody says important, that's important. Now, some people think that if other people, I think my problem is important, but most people doesn't agree with that. But I insist it's important because this is where I'm going to get my Nobel Prize. There are people thinking that way. There are a few of them. Mitchell, chemiosmotic hypothesis, right? When he proposed that, nobody believes it. Nobody cares. And he couldn't even get a good job, so he has to work in a research foundation. But he insists on that idea he thinks is important. There are people like that. But I think in most cases, 99% of cases, your peer usually decides what's important. Unless you have the confidence as Mitchell or confidence of Francis Crick or Richard Feynman, you should take seriously what other people think. You need to talk to people. When you're working on a problem, you talk to other people. What do you think? Is this important? Is this a significant problem? If everybody say, well, this is very interesting, this is significant, you gain confidence that this is significant. So you decide the problem is significant by interaction with your peer. I think that's the safest way, unless you really are a genius. You think you're a genius, you insist you are a genius, that's fine. But otherwise, take seriously the opinions of your peers. But no, there are many different types of important problems. I think there are 3 types. The first type is all your peers, if you're in your field for a few years, you know what's important in your problem. These are the problems that nobody can solve it. Now, in brain science, there are problems we know it's important. What is the consciousness? Things like that. Very few people work on it because there's no clue how to work on it. Right? What is the meaning of dreams? All these very important problems. It's hard to work on it. There's no handle. So this is how everybody thinks is important. That's true. But it's hard to work on it. But there's another type of problems everybody knows is important. It's clearly solvable, but it will take a lot of resources. Now, when the genome project appeared on the scene, people know it's important. Genome can be sequenced. But people disagree with whether you should do it at the time because it takes too much resources, take resources from other places. So you know it's important there. Whether you want to do it depends on whether there's the capability of doing it. Well, in brain science, there's also the problem of analyze the 3-dimensional structure of the neural network, complete analysis in 3-dimensional level. You can do it, but it will take 100 years or 100,000. I don't know. There's a calculation of 100 postdocs for 100 years. You will solve a small piece of your complete connectome, right? So it's a resources problem. So this is the second type we don't want to discuss. Most of the research scientists don't care about this problem. All we care is the problem that has some clue that it can be solvable. There are many problems like that, right? So that's where you choose for your research problem, where the innovation will be accomplished. It's those problems that are solvable. Now, there's this saying that science is the art of solvable, Peter Madoff. What it means is that science is an art of solving problems on the surface. And Peter Madoff says it's actually an art of making a problem solvable. You have to find ways of getting at it to solve this problem. That's the art. So in this range, this group of solvable problems, how to get it? There's no general rule. It probably is experience. Over the years, or from what you hear from your peers and what your supervisor would advise, you choose problems and choose a way of solving that problem. And in fact, another way of looking at this art of solvable statement is that it's an art of finding a problem. It's solvable. That's even more important. Most people choose a problem that's too difficult, so you cannot solve, but there's some clue that can be solved. But choosing a problem to work on is actually the key. So now that's what you have to learn over your career as a scientist. But I want to talk more about this selection of this third type of problem, where you know it's solvable, whether you want to choose that problem. It sometimes depends on how much risk you want to take. Are you allowed to take risks? There are many important problems we all know, okay, it can be done, but we don't do it. We shy away from important problems that could lead to innovative discovery. The way we shy away from it is because we cannot take risks. There's always an excuse. As a graduate student, you have to graduate. You have to publish paper in 5 years, in 4 years, in order to graduate. So if the problem is too difficult, you cannot do it. You are not allowed. Oh, no, a beginning investigator has to get their paper published within 5 years to get tenure. So the environment seems to not permitting them to take risk. That's always the reason you cannot attack the problem that is important that could lead to discovery. Now, but in fact, among all the same circumstances, there are people who made the innovative discovery, young people, young graduate student, a young researcher. They did it. Even though it's the same environment, same risk they are taking. That's because they are willing to take the risk, right? So given the same circumstances, the people who are actually taking the risk has the chance to get an innovative discovery because important problems that always there that it depends on when you take it. So I had a statement which I think is correct. I think many of our older people probably will agree. Over the career, I've seen many scientists. They have different level of accomplishments. I think the accomplishment of a scientist over a long period of time, I think is directly proportional to the amount of risk he or she is willing to take. And that's my conclusion. Right? Those who are willing to take risks over a long period of time, they are all more successful. More successful means more innovative discovery. Right? So the risk taking is very important. Even if you, at a condition you think you cannot take risks, but some other people can take it. Why can't you? Right? So in fact, the risk is overstated. The environment that doesn't allow to take risk is overstated. Now, in China, the government keeps saying that, well, no, you're allowed to take risk now. Well, you can apply for a grant, even if you produce nothing, as long as you're doing innovative research, you are supported, it's fine. Right? Government keeps saying that. That means that you are allowed to take risks, but nobody takes risks. Because it's a habit, right? It's a culture, right? That you need a courage, a habit of courage of taking risks. There's a word called audacity. Right? The audacity, the best. If I write comments or the recommendation letter for my graduate student or for my postdoc, I always use this criterion: whether if this guy who has audacity to take a difficult problems and solve it. Or even if he doesn't solve it, he has the audacity of doing science. And that's really the best, I think, best quality of a scientist that has the potential to become innovative. Now, if you go to extreme, it's called killer spirit. Right? The killer spirit is like, you got a problem, it's important, you have the spirit to solve it. Right? Most of the Nobel Prize winners, you look at them, they all have the killer spirit of solving their problems. Right? So that's where the innovation finally comes from. So besides this importance, every problem, even new, it's novel problem. It has a continuity. Now, every science advance, every innovative discovery is the work done on the shoulder of other scientists. It doesn't come out of the blue, right? Now, it may be not the shoulder of a giant scientist, but it's other people's shoulder. Now, you can see a lot of theories and proposal of very novel stuff by laymen. They all have a theory of how body works, how mind works. But they are crack science. They are not real science, novel science, because they are not on the shoulder of the working scientists. Right? They are not using the same ideas, language, or concept or methods that everybody scientists work with. So to be a novel is not means that totally brand new. You can dream of anything, then become novel. You can. So it is an important aspect, continuity. And the science advanced by revising or supplementing the current knowledge. So novelty is not completely new, but it's always an improvement, revision of the existing knowledge. So then the other property, which is a little bit more controversial. Now, the novel innovation in science is unique. Now, if you don't invent something today, somebody else will invent it tomorrow. So there's no novelty in terms of invention or innovation in science. Well, if Watson Crick didn't discover DNA, people say, well, Linus Pauling would come up within 6 months. His correct model would appear. Somebody else, Wilkin, would come up with the correct model. Right? This is the argument. So is science unique? Is innovative work unique? And I think this is debatable, but I would say, my opinion on this is that even if you have the same conclusion of paper in your work, the same conclusion can be reached by different route, right? So it's not the conclusion of your finding or the conclusion in this case, in most cases, conclusion of your paper. You announce your finding. The conclusion itself is not the only thing. It's how you reach that conclusion. Now, different people, even if you have the same conclusion, they reach there by different experiments, different evidence. So that's unique. I think that's different. Now, the conclusion can be supported by different degree of evidence. Now, some people can have a paper published 20 years ago, the same conclusion, but nobody seems to care until recently you publish a paper showing the same conclusion. Now that old guy comes here. No, you are just making the same conclusion. What's new, right? But the reason that paper 20 years ago didn't get people's attention, people doesn't pay attention to that work, is that the evidence provided by that, even if there's a conclusion, the evidence underlying is not good enough. So it doesn't have the impact. Right? So it's not just a simple conclusion that justifies the innovative discovery. As to how much evidence you have for that discovery, and how do you do it? How do you support your evidence? So I think that that's where the uniqueness comes from. Now, different way of presenting your evidence is also different. The argument you make is different, and the conclusion, even if it's the same, there could be a different style of presentation. So every paper, even if you have the same last sentence in your abstract, the way you get to that is different. So that's why I say every paper is unique. Now, I think each scientific paper is as unique as a piece of artwork to be appreciated and enjoyed. Now, to stress that point, I would give you an example of this most famous paper in biology in the 20th century. It's the 1953 April 25th Nature paper by Watson and Crick, describing the molecular structure of DNA. Now, this is one-page paper, right? In this paper, they propose this DNA structure, right? This anti-parallel twofold symmetry with a particular helical pitch. A conclusion they reached by model building based on X-ray diffraction data by Rosalind Franklin. So that it's a correct solution. They reach it by reasoning, by model building. So they wrote this paper quickly within a week, and ready to be sent off to publication. Right? They know it's going to be published. And then they thought about it and said, well, we have to say something about the implication of this structure. Right? You just have a structure, what does it mean? How important is it? No, we got to say something about it. So they, at the end, before acknowledgement, they insert one paragraph. And I think this paragraph tells you this most important paragraph in biology in 20th century. This paragraph says, it has not escaped our notice that the specific pairing, this nucleotide pairing that we have postulated immediately suggests a possibly copying mechanism for genetic material. Right? The way they present it, it's kind of British, right? It's sort of reserved. It's not, have not exposed. We notice it's noted. We have not escaped of notice. And this the most important thing in the genetics is the copying mechanism. Right? So nobody else is going to write this way. Linus Pauling certainly will not write this way. The entire paper, one-page paper, even if DNA is discovered 6 months later, it's not going to appear in the same way. That probably Linus Pauling already had a paper of triple helix. He couldn't blunder about maybe they produce a model which is still wrong, not completely right. But then another paper comes in. Incrementally, you finally reach the final solution. But nothing like this, a single page, complete description of a structure and its implication in such a concise manner. Right? It's another artwork of scientific literature. So I think it's unique. Now, you cannot say that Watson Crick, if they don't discover, other people discover it in a different way. So that I say it's unique. So, and also the innovative work has an element of completeness. It is complete like the DNA paper. It's complete in presenting all the essential evidence and the essential detail. It's not everything. It didn't talk about everything of their finding, but the essential structure is there. It's like making a sketch of a face. Right? The sketch, the good painter quickly have strokes of important strokes that would reveal the entire face, the essence of it. That's complete. You don't need to detail of all the face to paint it in detail. So essential detail, essential evidence of your discovery is important. That completeness is important for this truly innovative work. It's complete also in the covering in your usual paper. It's better that this is a complete work if it covers all the possible alternative explanation. If you have experimental evidence to cover it, it's fine. If you don't have experimental evidence, you at least discuss about it, so that the reviewer will not come back and saying that, well, you didn't think about another alternative. Right? That's a sign of your finding. Your paper is not complete. You have not discussed or considered other alternative explanations. So completeness include the interpretations. Now, there's another property of novelty, is the timeliness. This is very important because science is very competitive now. The work that appear at the right time in the right place take notice, have the largest impact. Now, there are work that appear somewhere. It's important, but people didn't notice. So there's this famous Mendel's discovery of the rules for inheritance. That's premature, 40 years earlier, too early. Is there a discovery like that? Possibly. Right? But in these days, this type of premature discovery, if it's important, it always gets noticed because the communication is so widespread, so easy to get the information. So this comes to the problem of scientific communication and publishing your finding, telling people of your finding, It's the way to announce your discovery. That's a communication. But when do you announce it? It involves the element of competition, right? People always argue whether you should talk to people about your unpublished finding, because before you publish it, if you talk about it, other people knows about it, they may publish earlier. You'll be scooped, right? So then your timeliness is lost. But if this is the case, why should we have conferences? Why should we communicate with other people? You shouldn't present your poster in the big meetings because those are not published yet. If you present it there, you have a chance to be scooped. Other people will take advantage of you by knowing your result. But why there are 40,000 or 30,000 posters in the Society for Neuroscience meeting every year? Why people still do that? Right? It's because on the whole, in the long run, telling people what you're doing has benefit. It's beneficial in the long run. It's beneficial in several ways because you're standing on a poster, even though your paper not published, but you're telling people early, say, I have this finding. People will say, well, this finding has problems, and you've missed this experiment, that experiment. You get information early before you really have it in print. Right? That's the biggest advantage. You announce it early in an unpublished form. That's timeliness. People know, oh, somebody's already done this. If it's fair, people in your field, some fields are more competitive than others. In some areas, people are fair. People knows who have done this first, who has discovered this early. I mean, if you announce in a poster, you'll be recognized. Well, on the other side of this story is that this is the opportunity to tell people that I have done it. I have this discovery already. You don't need to bother to do it anymore. Right? You claim the territory by giving a poster early. That's another way of competition. Right? So it is work both ways. It is because of this you talk about unpublished findings. People tend to shy away telling other people what they are doing. But I think on the whole, this is important to consider. So now, this is the most common sayings that you hear in scientific circle. I got to publish this paper, otherwise I'll be scooped, right? The timeliness. Now, but if your work end up to be scooped, what do you blame, right? Somebody else is a bit earlier than you've published your finding that you are just working on, you are about to publish, right? I think most people will say, well, somebody has stolen my ideas. The last poster, he seems to have visited my poster. That's why he got the first. People tend to say story to you. When you are a young people audience, don't believe that story about being scooped. Most of those stories are exaggerated. Including your supervisor telling you the story that he has been scooped is probably exaggerated. Because in most cases, it's not due to unfair competition. It's just that you are just a bit slower than other people. Ideas are all there. Everybody knows the important ideas, important problems. Now a lot of people are working on it. It's not you, the only one who had the idea, right? So there are competition that just seems to be getting there earlier. That's it. That's most cases of so-called being scooped. Now, but how do you balance whether to send out a paper quickly so you don't get scooped? Well, that's a decision every working scientist has to make. Right? So you have to judge between the completeness, which I just mentioned. You want the completeness of your paper that has impact. And in the meantime, you want the timeliness to send it out. So how do you make the judgment? Different people make different decisions. But my point is that because of the scientific publication have the uniqueness I just mentioned, being scooped is not the end of the world, because every paper is different. Now, your evidence and those people in a published paper are different. And even if you cannot get your paper into Nature and Science, but you will get a paper, if it's a solid paper, in a good journal. In the end, it really doesn't matter, because really your work could be more substantial because you're a little bit slower. Even though that paper was published in Nature and Science, the evidence not as good as yours. People in the long run, your paper will have more impact because your conclusion is better supported by your evidence, right? So this is, your work can be more supported and contribution will be more valuable in the long run. Now, this is not just the way I am just putting this out to you as a way to convince, to support my argument that you shouldn't be too worried. I have my own career, have the evidence. The best paper, the most cited paper in my entire career, it's not a Nature Science Cell paper. I published many of them. It's a Journal Neuroscience paper. The Journal Neuroscience paper is a trade journal in your field, in my field. It's like a Journal of Virology, a JBC, Journal of Biological Chemistry. This is a trade journal in your society journal. It's the most cited paper because the reason that paper was published late, because there was an earlier paper in Science that essentially have the same conclusion. It's Bert Sackman, Henry Markram has that paper. But we did it much more thorough work. We had characterized more detail. This is called spike time-independent plasticity. I can talk more on that on Thursday. This finding had much more influence, much more impact than the earlier paper in Science. People cited more because it is more solid. So I have my personal testimony, I guess, is what you call it, that being first is not everything. Being complete, being thorough, compensates for your being late. So this is what I want to say about timeliness, which I hope you remember this, especially students who are going to have their paper being scooped by others. So how do you learn to be innovative? I have to keep track of my time. The origin, where's the origin of innovative idea? Where's the origin of imagination, creativity? Now, my simple answer to this, the origin is not coming out of the blue. It's not inborn. It's something, ability you learn. So now, I think to be innovative, if the way to learn to be innovative, you go to the history. You need to spend a lot of time exploring the history behind innovative work, in your field or in other fields. Well, why is it? Most people read a paper, right, or know a finding, or innovative finding. You know what it was discovered, but you need to know who and when and where, and why that discovery was made. Now, this 5W is what you need to know about the history of an innovative work. Now, why do you want to know this history? Because if you want to make innovative finding, you need to know at a particular time, particular space, making a particular decision to do some experiment. Now, where does that idea come from? You need to learn from what people have done this before, so that when you are in a similar situation, you make the right decision. Now, not exactly the same choice you're going to make, but the way that the innovative work was done will give you this thinking, will shape your thinking of what you should do at certain circumstances. Right? So knowing the personal history of the innovative discovery is a way to learn to be innovative. Right? You learn from those who have done it. The history is always missing, often missing in the textbook. There's very few textbook tells you the history of how DNA structure was discovered. Right? You have to read Watson and Crick's autobiography to figure out how it was discovered. Right? So, and most papers has a distorted history. Right? In the current paper, now there's a scientific bagu. You always started with what has been discovered before, and now I propose a hypothesis, and then I do an experiment, I prove it. And then that's it. Now, in the end, in reality, nothing, very few cases like that. You stumble into some findings, and then you figure out how do I explain it, phenomenon. Then you propose your hypothesis, and then that seems to make sense. That's how you write your paper. And so the history is distorted in the current scientific literature. You cannot find that out. You have to go to personal history. Right? So how do you do that? So I suggest that next time when you have a distinguished lecture tell you about innovative work, you have standard questions to ask them. And people should always ask questions afterwards, not about his work, or the detail of his work. You should ask the standard question to an innovative scientist: how did you come up with the idea? Now, he usually doesn't tell you in his lecture. And why did you do the experiment at time? These questions that the visitor in a short seminar summarized in years of work never tells you about it. And if you ask this question, this is a universal question for everybody, right? If you're short of questions, just use this question. Once you ask the question, you get the best answer from the scientist. It's worthwhile. He will tell you, no, the original started doing this because my graduate student had this weird finding. Now I look at it, now maybe we should do this. We should pursue this idea. So we then do the experiment, we get this and that, right? So this is a story totally different from the very logical presentation you hear in the standard seminar presentation. So remember this question, right? Ask that to the innovative scientist. This is how you get a history behind the innovative work. So there's the best book in biology about the history of molecular biology. How this revolution of molecular biology comes about. It involves 100 scientists over a period of 20 years. This book is summarized in this book called Eighth Day of Creation by Horace Judson, who is a science writer, used to be a science writer for Time magazine. He read all the papers, he interviewed all the scientists, asking the question that I just proposed. He has tape recording and then summarized that. Tells you how did the molecular biology revolution comes about. So you have a documentation. You want to learn to be innovative, you read this book from the beginning to end. More important, it's a very thick book. If you don't read half of it, you'll become half innovative. If you read it completely, you'll be completely innovative. Right? So this is a book I recommend to my graduate students, not to take a course in molecular biology. They should read this book rather than take a course in molecular biology. So learn to take information selectively. This is very important to become innovative. Now, there's a crisis we are facing, the crisis of information overload. There are too many papers, too many scientific information that we need to go through our brain. So too much information is bad. It's confining and inhibitory. Now, why is this? Because this has to do with, now there's no brain theory for how innovative idea comes from the brain. But if I want to propose a hypothesis, I would propose a hypothesis of a neural circuit loading of information. If the neural circuit is responsible for your thinking process, if this circuit is constantly being loaded with information that store the information about how thinking process is going to be, then next time you think about a process, you are going to think about what you have read. It's other people's idea. It's no longer innovative, new ideas. So having your circuit used for storing information, if it's too much, it becomes confining and inhibitory for new ideas to come up. So people always say that young people are more creative. We old people are less creative. Now, it's not because my brain, I have a neurodegenerative disease, my neuron is dying. I think it's because my circuits are overloaded with information. Every time you come up with ideas, so-and-so have done it 15 years ago. And then that doesn't work. And you come up with another idea, some people think that's how it works. nothing, it's all this information already loaded in my brain. I cannot get rid of it. And when you heard the 出生之犊不怕虎, the young bull, young cow, doesn't afraid of, it's not afraid of tiger because they have never seen tiger. They don't know tiger can eat them, right? So 出生之犊不怕虎. That's why young people can do science, no innovative science, because they don't know it doesn't work. It doesn't work that way. That's why they can try it. They would try something that eventually become innovative, right? So don't take too much information into a brain. So I have warning to beginning researchers: do not read every paper in your field. That's the best way. That's the best way. The worst situation for a beginning student is download all the paper in your field, 100, 200 paper, and spend a month reading those. Right? Because you have no way to judge whether the finding is correct or not correct, reliable or not. Right? You have no experience in the field. You load in all this information, is totally inhibitory for your thinking. And you can read. You have to read. You read good reviews. You read good reviews in your field. It will tell you what are the known problems that has been solved, what are problems that's unknown, unsolved. Then you just do your research. You do your experiment. When do you read the papers? When you get your own data. When you have your own data, you have done some experiments. You come back, you say, well, I have to write a paper, then I have to cite all the earlier papers. So then I'll go back to literature and read it. You find out, oh, that paper I don't even want to cite it because I know that technique doesn't work that way. I have experience to judge. You can reevaluate your literature. Before you have the ability to evaluate this literature, don't be swamped by this information. Most of them are wrong anyway. So I have a suggested sign, warning sign on every computer sold, every package of cigarettes sold, that too much excessive, non-selective reading is hazardous to your health, to your mental health. Now, the other thing you have to do to be innovative is to pay attention to seemingly unrelated fields. Now, there's over-specialization in all fields, right? So we are trained in a very specialized area. Now, there's limitation in our graduate school training, laboratory training or curriculum training. They are all very limited to subspecialty. Now, you always go to a seminar. Most people doesn't go to seminar unrelated to that work. That's wrong. Because go to seminar unrelated work is where you can really learn innovative idea that can be applied to your field, right? So in fact, you should always go to, put high priority, go to seminar unrelated to your work rather than related to your work. That's where you should go. Because the things that relate to you, you already know something about it. If you really want to know more, just read the paper. You know where to find the papers, right? But something remotely related is what you should do. You should talk to people outside your field, right? So now, when I visit a place, I always want to talk to people that I don't know, unrelated to my own work, because those who I know related to my work, I already know their work. I don't need to visit, that much. It's talk to people outside your field is more important. If you want to find a postdoc, don't do the same thing. You go to an unrelated field. Now, it's hard to get an unrelated field because they don't hire you because you are totally unrelated, right? So you need to convince. If you work on it, if you find an unrelated field that you are interested in, if you work on it, you can get in those labs. Right? You need to be persistent. You need to ask your supervisor to write good recommendation letter. Eventually, you can get in the field unrelated to what you have been trained to do in your graduate training. And that's very important. Right? You opens you up. And in fact, in that different field, you actually could probably get more out of it because you have a different training. So you'll do experiment different from people in that field. And you have more novel, innovative finding. So now when you reach the time to get a job, you should find a research job outside your field. Now, people is afraid of changing fields. I'm training in computer science. I don't want to do a job in non-computer science related, in brain science. But I think the better career choice is to get a job not in your field, but you use your training to do that, do the work in that field. You look at all these new innovative ideas, nanoscience especially. All the people from different fields are getting into this problem and make an innovative discovery. Right? Because you have different training, you have different contributing people from chemistry, from engineering, all do this molecular nanoscience. Right? They are from different fields. They have different innovative approach. So if you want to develop new career, new research program in your lab, when you have a lab, no. Develop research program outside your own field. Because in those fields, your technology, your technique, your thinking will probably make more headway than those already existing in that particular field. So changing fields should be a norm rather than an exception for career planning, for future development of your research career. Well, that's my feeling. Now, this is very bad, especially in China. The undergraduate training is 专业, right? You go into a 专业 means you're a professional. You're a professional in that particular discipline. That's wrong. In fact, you should go away from that 专业, right? So changing 专业, right? It should be a norm rather than something you're afraid of. So the next thing is you want to discuss and to be innovative, you want to discuss science, describe science orally, frequently. Now, it's very interesting that you don't do science alone in your office, talking to nobody. That way of doing science is very ineffective because talking about science with other people is very important. Talking is a process. Describe science is a process of logical integration. But all the information you have obtained, by talking, you actually integrate the information. You have to, what talk first, what's the second. You have an integration of the existing knowledge into a logical sequence that you can present in your description. And that training is very important. Right? This is very important. Therefore, for example, having discussion, you would have debates. And you take an argument, people talk to you and disagree with you. You have a debate. The debate and polemics, right? Counterpoint, arguments is the way of coming up with new ideas. This is how innovative it comes about. You know the story that Watson, after Watson and Crick discovery, the new character appeared in molecular biology, Sidney Brenner. He substituted Watson to be the person who talked to Crick. Right? In the Cavendish lab, or the later molecular biology, MRC lab, Brenner and Crick share an office, and they were so famous. Now they can have a huge, nice-looking office. They don't want it. They want to share a small office together. For 25 years, they have the same office. Why? No, according to Brenner, they love it. All the ideas comes from talking. Right? They always argue with each other. They are sounding board, right, for ideas. So when you argue about your ideas, you are innovative. You come up with a way of that support your ideas. What is evidence I can support my ideas? This is the breeding ground for innovative ideas. That's why the talking is so important. Now, this effective communication or talking requires practice and training. I think the most important thing for graduate school training is to be able to present and discuss science, not just writing papers or finding. Right? If you didn't learn the ability of talk about your science logically, effectively, you didn't have a good graduate school training. And much of the joy comes from communication, talking. If you want to have a scientific career, you'll know that the most enjoyable thing is to tell people your finding. That people say, that's interesting. Right? That's wonderful. People will appreciate your findings by communicating, talking. Right? So I have a colleague, Gunther Stan, some of you might know, is an old molecular biologist. He has passed away now. When Gunther was alive, we have dinner together. Gunther would once ask people on the table, now, would you still do science if I give all the resources you want, all the grants you want, equipment, whatever you want, but you are forbidden to talk to other people. You have to do this alone in an island, somewhere in Pacific. Would you do science? I asked people, I'm a scientist. No, nobody would like to do science because if I cannot talk to people, who knows my finding? Where's my glory comes from? Where's my satisfaction comes from? Right? I cannot talk, I cannot publish. Then why should I do science? Right? So that's tell you how important is the communication is for science. Now, next, you have to pay attention to unknown. Stuff in the textbooks are incomplete and questionable reliability. No, textbook doesn't tell you what's unknown. It tells you what's known, right? In the scientific literature, usually they don't tell you what's unknown. I mean, they tell you what the discovery was. So this was a statement by a biochemist when they give a talk, give a lecture in the biochemistry course in Harvard. She said 50% of hypotheses in text were wrong, but I don't know which 50%. You better be careful. So that's why the status of the field. So you have to know that a good textbook, a good review, is the one that tells you at the end of that description what is unknown. Right? That's to me a judgment of whether it's good or bad. Science progresses by revising existing knowledge. Finally, you need to learn to self-educate. Now, it would be nice if my advisor is a Nobel Prize winner. Then I'm sure I'm going to do great science because he's going to give me good ideas to work on. But I didn't get a Nobel Prize winner as my supervisor. Very few of us had that opportunity. But if you read those papers, those books, well, Watson, Crick, they all tell you how they become innovative. They have all kinds of books written. I know this story about how DNA was discovered, or how various discovery in our field, in my neuroscience field, was done better than their own students. Right? Because I read all their books and I can memorize them in heart. So I can educate myself. I can take 100 Nobel Prize winner to be my supervisor, right? To be my advisor. Right? So self-education is important. So there are many books. If you are interested, you write me an email. I'll send you a list of books I think is very important. This is mostly for biologists. I have commentary about this book, so I can send it to you if you're interested in. Most of them are the history book about science. Now, if you don't like to read, I mean, most people just want to get onto the website. I have a website for you. It's called the Biographic Story on the internet. It's called Web of Stories. Now, this is the best science website. What it consists of is the monologue. That is an interview of famous scientists, all the biologists you know, important biologists, physicists, mathematicians, chemists, even including poets and architects and all other fields. Every one of them gives you hours and hours of monologue about how their history, how they made the discovery, what their life history is about. When they reach the difficult time, what do they do? When they have a decision to make, scientific decision to make, why didn't they make that decision? Right? So if you have 5 minutes, 10 minutes, you can get onto this site because all these monologues have subtitle. You want to go into one region. In Brenner's autobiography or monologue, it's 5 hours monologue. Now they are cite you on Crick, on Watson. He'll tell you a story about his thinking about Crick and on Watson. Right? RNA, how they discover mRNA. Now, there are small stories you can pick and hear talking for a short period. But I warn you, if you are interested in science, if you get onto this site, it's very hard to get off. Let me see. I think my time is up. It's already 1 hour. Any limitation? 10 more minutes? 10 more minutes. I want to quickly talk about two approach to doing science. The exploratory research, right? Where you aim at discovering new phenomenon. People say, well, we're fishing expedition, right? That's the general description. You go and look for something. You don't know what you can find, right? Find some gene, find some phenomenon. Now, the exploratory research, in fact, are guided, right? This is guided by intuition, imagination. This is all very mysterious thing. Random thoughts, right? You can do this exploratory research. But where are these come from? Now, according to what I'm saying before, these are not random in a sense. This source comes from a seminar you have heard, unrelated to your field. Something that somebody has said during your communication with talking. All those random thoughts comes from somewhere. Right? So this is exploratory research. It also requires experience. Right? The experience is the name everyone gives to their mistakes. Right? So your experience always don't shy away. Don't feel mistakes are failure. That's how you gain your experience. Right? So in exploration, you'll find a lot of failures, but no, that's experience. Boldness. I mentioned that you need some sort of boldness for exploration. And people say, well, the exploratory research are random, just fishing activity. You just go there and fish. You're lucky you'll get something. But they're always guided somehow, a vague hypothesis. You have some thinking in your mind that what you're going to find. That's how you do your fishing. You know where to fish. That's how exploratory research needs to be. I'll make an example for those biologists that know this example is the discovery of cell reprogramming. Now, Shinya Yamanaka, who discovered the iPS cells, induced pluripotent stem cell, who gave a talk here, I guess, a couple of years ago. This very important discovery. Now, you say, well, his fishing expedition, he doesn't have any, he's explored all the transcription factors that can reprogram the cell. They happen to, after a few years search, he got that's 4 factors that works, right? This is a typical exploratory research. But I can say that he's guided because he's also guided by the idea that the transcription factor involved in development are likely to be the transcription factor that reprogram the cell. Right? He's not randomly test all the transcription. When you do random combination of this, he's guided by vague hypothesis. You reprogram him by changing the combination so that you can push the differentiation in the opposite way. Now, this is another good example is Doug Melton. After a few years, when the iPS cell was discovered, he has a vague hypothesis that, well, if you can reprogram him from differentiated cell back to undifferentiated cell, why can't I just reprogram a differentiated cell to another differentiated cell without going backwards to the origin of undifferentiated cell? Right? It's transdifferentiation. Well, the vague hypothesis is very clear that if you want to do this, it must be some transcription factor that involved in this alpha cell and beta cell, 2 different cell types in the pancreas. He switched alpha cell to beta cell by transcription factor, a set of transcription factor. It's not a random search, it is exploration, but he has an underlying basis, is that the factors, transcription factors during development that are responsible for the beta cell differentiation are likely to be involved. So when he do this sorting, it was done one of his Chinese postdocs for 2 years of combination of factors. He actually was guided by some ideas, right? Even though this is exploration. So one can ask one more step. This hasn't been discovered. Some people, I think within a year or 2 or 3, somebody would come up with a discovery that in vivo cells can be transdifferentiated. Nobody has done this, have shown this. The transdifferentiation is you transfect the cell with a certain factor, they switch to different cell type. But if this is possible, why can't the cell in vivo, when it's needed, to transdifferentiate into a different cell? Why can glial cell change into a neuron when it's needed in some site of injury? Right? That's useful. Right? So this is a hypothesis, right? Then you started you can start let's say, exploration. Look for factors that could perhaps associated with some cytokines that are induced during injury that might be transformative of glial cell into neuron. Right? That's possible. Right? Everything is possible as long as it's not violating physical chemical principle. So the key to exploratory research, there are many things you want to do. You want to maximize the parameter space of your exploration. You want to cover border space. And also, a lot of exploration takes a lot of time. So the virtue of quick and dirty experiments. That you don't want to do very detailed experiment when doing exploration. This is actually what Julius Axelrod, who discovered norepinephrine, who said that all his life is doing dirty and quick experiment because he explored. Right? The way he explores, he covered many different possibilities without spending time to get more very precise experiment. Once they get something, then he start to do more precise experiment. You always, in our exploration, you need to optimize the parameters before you, once the effects observed. Some people are very quickly, that's the problem of my graduate student, that's why I put it on. My graduate student find something, they immediately want to do next experiment. So I always try to slow them down and say, well, why don't you get the effect bigger, more robust, before you go further? Right? When doing exploration, tend to say, well, I discover something now, then I will do the next step. Right? This is often a problem of doing research. And in the end, when you do further, you find that this effect is too small. After you do a long series of experiments, it turns out it's too noisy. Right? Because when you initially you didn't optimize your parameters. Make sure the reliability of methods, positive control is very important. Make sure that reproducible of effect. Somebody else, your friends or lab mates should repeat if you have some important discovery. Well, so this is what I said. In biology, you propose, there's a hypothesis-driven research. I don't want to say too much about it. What is a good hypothesis? A good hypothesis is a hypothesis that really gives you some pricing elements. You propose something, people would raise their eyeballs. Oh, I haven't thought about this. If you propose a hypothesis to somebody, you say, oh, of course, that should be true. That's not a good hypothesis. You want a hypothesis that has a surprising element in it. That's where to lead to novel hypothesis. Right? This is a good hypothesis, a novel hypothesis. A good hypothesis also have very clear causal relationship. That's a testable relationship. Hypothesis there to be refuted, not to be proved. If your evidence doesn't fit with your hypothesis, you're always ready to modify your hypothesis. Right? This is very basic thing for hypothesis-driven research. If we have time in the future, I can go further. But I think the take-home message is in exploratory research or in hypothesis-driven research. I think in biology, any mechanism you can imagine, that's where the imagination plays a part. As long as it's useful to the organism and does not violate physical and chemical principles. And this mechanism is likely to be used somewhere, in some place in the organism, because evolution has such a long time to take advantage of all useful mechanisms. So that's the nice thing about biology, that you can just dream up a mechanism. If you're interested, if the mechanism is useful, and you probably would find it. This is just like the transdifferentiation, the endogenous, natural transdifferentiation. It's a hypothesis I think is useful to the organism, but it hasn't been discovered. So I think that should be somewhere. You just take us to find it. Right? Take some work to find that place. Now, when I thought about this, I was thinking that somebody must have said this, because all my ideas come from reading other people's books or talks to other people. So somebody may have said it. So I want to give credit to people who thought about this. And I looked around and I couldn't find it. So before I find this origin of this idea, I will claim that's mine. Okay, I think I should pretty much stop. There are other things. Now, you know this word, right? 大胆假设，小心求证。Be bold in hypothesizing, meticulous in searching for evidence. 有一分证据不说两分话。Do not claim more than your evidence says. Do you know who said this? Anybody? Hu Si. Yeah, because only old people knows about it. You young people should read about Hu Si, right? The most important philosophers or historian in our last century, Chinese history. Anyway, I'll stop here and thank you.
Thank you so much, Mu-ming, for most inspiring and illuminating talk. Do you take questions?
Oh, sure.
Okay, maybe.
Don't ask me where I have the idea.
Maybe a few questions from the audience? Maybe I'll start. So, in order to nurture and support innovations, especially in a university setting, do you think promoting cross-disciplinary research would be an effective way to do that?
Well, one of the things useful is to promote talking. I have a strong feeling that by talking, by debating, by these arguments, you have good ideas coming out of that. Right? Innovative idea. Now, I think I have some ideas by talking to Carl today. That seems to me, I haven't thought about it, but by talking, I think that we have some good ideas that came up.
So somehow we have to apply grant for our research, but it affect our innovation ideas.
You have what?
Apply grant for our research, but in this case we have to fulfill some of the community ideas rather than our own or innovative ideas. The grant actually, the money control our
Well, you're talking about teamwork versus individual research. So there is this conflict that if you join a team to work on a project that's defined by a group of people that your project belongs to, you somehow suffer the individual creativity and innovation. You don't work alone. Right? The idea that a scientist work on his own bench and come up with a brilliant discovery, this is possible, but in these days of science, it's very unlikely. Most of the good work, you see all the innovative work that comes out in every field, it's all involved more than one laboratory, more than one person. Teamwork always is the way that important problems are solved, because the individual lab just doesn't have enough capability of solving important problems. That's why your project is part of the bigger one. And you contribute to this teamwork by providing your unique contribution that could still be innovative. Now, joining a big project, have a common goal of solving a problem. Let's say, find the Alzheimer, the marker for Alzheimer's. Now, there are different approach. Now, there are genetic approach, there are functional approach, there are molecular markers. You are one component of it, but in your component of the teamwork, you still have your innovation. Right? A good teamwork doesn't hurt innovative individual creativity. Now, this comes with another problem with our system of evaluation. our current system of evaluating a researcher needs to be changed. Now, in physics, you can have hundreds of people working on discovering the God particle. But each one of them all are indispensable in one component of their work. The evaluation of the people who are accomplishing a group work are well established in high energy physics. They know which team, which work, what do we contribute to team. There's an expert evaluation of the contribution of each individual scientist in teamwork. They get their tenure, they get their support, they get a promotion, not based on their individual unique finding that other people have accomplished, but on their contribution to the team. That tradition in a large, big science is now spreading, should be spreading into other areas of biology which require teamwork. Right? So don't be shy away from this big project. And the biology system, in fact, in our center, in the CAS center that I'm trying to promote, our evaluations no longer follow the system, American tenure system evaluation, which is saying that you have to have independent demonstration of independent work to get your tenure. We don't do that anymore. You have a paper that's all co-author paper. You're all contributing 1/3 to this, 1/3 to that. You get your promotion and tenure because we can evaluate your contribution. And the evaluation system have to go along with the problem in order to solve the problem you are talking about. I hope in Chinese or in HKUST, some sort of consideration of how you evaluate tenure decision. Include, give more, don't divide it in half when there are collaboration of important work. Right? It should be 100% for both investigators. I don't know.
No,
Carl.
exactly. I'm looking at Carl. Change the system, okay.
Tsinghua University, I talked to the vice president, Xie Qikun. They said that they are starting. They think this is a great idea. They're going to do that for their biology new faculty. Their tenure decision evaluation. And a lot of places are in the past are dividing. Now you have 2 corresponding authors divided by half, 3 corresponding authors divided by 3. They don't do that. You have a good committee, you decide it. If your contribution is indispensable, it's absolutely required for that final product, you have 100% contribution. Tsinghua is doing that. Our CAS Center is doing that. Now, I hope that in the long run, other places will do that.
Because I think this would be a good way to promote collaboration.
Collaboration,
Yeah, because
teamwork.
I think a lot of PIs shy away from that because they don't know who would have the credit in the end. So we'll try to promote that here.
Okay, great.
Okay, and yes?
Yeah, I have a question. So it seems like encouraging risk-taking is a very important part of innovation.
Right.
And I'm guessing, I'm just wondering, like, what are your views on how you encourage risk-taking? Is it monetary, prestige? And also, how does that play into, like, the tenure system? So that is a system that protects sort of like academic
So the
freedom.
environment, you have to have an environment.
Do you think there's a better system for risk-taking at the PI level? And also, how does that affect us at the research level? How do you encourage
But the environment or the system should encourage that. Together, not just verbal encouraging, you should take risk. Nobody's going to do that without some inducement. The inducement is, for example, in our own institute, our tenure decision is 8 years rather than 5 or 4. Right? I say, well, we 8 years tenure review, twice review, 4-year review. The first 4 years, mid-career review, no decisions made. Even if you have no publication, you are taking a risk. As long as the project is evaluated as a good project, potential good project, then you just encourage to continue. Even if you are considered not a good project, you are still allowed to stay. If you have nothing accomplished in 4 years because you're taking a risk, you are not asked to leave after the mid-career review. It's an 8-year review that gives you a room. You say, in 8 years, you got to show something, that your idea worked. Your risk-taking has some accomplishment. Right? So that's the sort of environmental, the institutional structure that's required to provide a risk-taking. The other thing is the funding system. Right? The funding should be encourage a project that are risk-taking. Now, this is actually the bigger problem, I know it's probably here in RGC review, is that if you have proposed something that's too novel, taking too much risk, the reviewer will come back and say, this is too much fishing expedition, this is too risky, it's not safe. Right? They will come back with this and you didn't get funded. Right? So this is the system, review system. The reviewer has to allow the risk-taking project, or novelty aspect.
Okay. Yes, please, Ken.
Right.
So I think that innovation has some elements similar to creativity. So there was a saying by Ken Robinson saying that actually education kills creativity. So I want to hear about your comment.
Well, that's too broad a statement. Now, people have to educate it. I mean, if you don't, education is important. It depends on how you educate it. Some education kills, some form of education kills creativity, I think. Right? So it's how we all know how we want to improve the education system to allow more creativity. Right? So the university education, graduate school education needs to be improved. Now, primary school education as well, right? From the very beginning, how do you encourage innovation, creative ideas, even at the primary school, middle school level? So it's a problem for educator.
So therefore, the reverse question is that do you think that if we see the younger generation when we comment that they're not innovative enough, maybe the system is the problem, the other part are the educators. Another problem.
Well, so the system, including the educator, together, right? So that's a problem, right? That should allow. But no, to be innovation is not just a slogan of 万众创新. You got the innovation, right? No, it's not slogan. You have to be. In fact, I don't think that statement of 万众创新 is actually very useful, because you cannot, if you don't get the training, you cannot innovate. You have to have a basic training so that you can make innovation that's relevant. Now you make a small gadget. Now everybody makes small gadget, and we will never have important innovation in our society. Right? So that's the problem of the assistant and educator. Right? How do you promote innovation?
Last question.
Now in Europe, China, have this big science project.
Right.
So what's your view on it?
Right. So the big science project, you're talking about brain science project. Brain science project. So there's the China Brain Project is coming. Probably in a couple of months you'll see an announcement. The China Brain Project is a reaction to the wave of interest around the world, global interest, from Europe to US to Japan. Every country has a brain project, big brain project. Now China has to do it. So the Chinese Brain Project, after 2 years of discussion among many people, come up with a much more comprehensive plan than the other brain project. You know the American Brain Initiative is a technology development, new technology development. European Brain Project is a supercomputer simulation of the brain, very technological oriented. Japan Brain Project is a project on using marmoset as a model system. Entire projects on marmoset. So these are not a project that cover everything in their country, brain research. Right? They are a small portion of the brain research. it's spearheaded new directions. China Brand Project would be much more broad-based. Briefly, it would be a central core of basic understanding of human cognition, cognitive process, neural basis of cognition, and with two applied arms, one on the brain-machine intelligence technology. Now, brain-machine interface, brain-like computation and devices, brain-like robotics. Then the other side, brain disease, early diagnosis and early intervention. So this will be the scope. And I think most people would agree when look at this, the Chinese Brand Project would be a project that has very selective, even though this is a broad base. It will be selective into area that are important for society needs. The central core is also selective. It's talking about cognitive process, especially higher cognitive process. Right? So the result is that you have a China Brand Project, which is addressing international frontier problems that everybody wants to solve. In the meantime, there's some advantage that Chinese Chinese project can be leading, we're leading. For example, the use of primate as a source for model, the use of large amount of patient data for brain diseases. Right? So there are uniqueness in this China Brand Project that take advantage of the unique resources in China. And I think that in the long run, in this unique area, that China will be leading in the world. Probably not in this high technology development, which is China is way behind the US and Japan and Europe.
So Professor Poo will be here until Saturday. And so if you would like to have a chat with him, please let us know. So again, please join me in thanking Professor Poo for a wonderful talk.
Thank you.